Okay. Okay. Good morning, good afternoon to all of you joining from various parts of the world. My name is Vinu, and I'll be chairing this session. It's a real pleasure to be here and chairing this session. Thank you to Karthik for the kind invitation. So, as we all listen to the opening session, so it is very clear that microplastic research is actually gaining importance, and there are like multiple facets to it. And this first session is really geared to provide a very fundamental and understanding about the analytical techniques that are available, that are upcoming in order to understand the quality and the composition of the microplastics in various environments. So we have two keynote speakers and two invited speakers in this session with a good blend of analytical Py-GCMS technique and spectroscopic techniques. So with that, it's a real pleasure for me to introduce the first speaker to you. The first speaker is going to be Professor OHTANI-san, who's a full professor at the Graduate School of Engineering, Nagoya Institute of Technology, Japan. He has published more than 220 papers and has extensive experience in the area of analytical pyrolysis, GC mass spec, applied to a variety of materials, including plastics and microplastics. He has served on the advisory board of the Journal of Analytical and Applied Pyrolysis. In the microplastics area, he's been working for more than 10 years. I think this is a real opportunity for us to learn from Professor OHTANI-san on the advancements in his research. Over to you, Professor OHTANI.
Thank you very much, Mr. Chairman. So I share my screen. Okay. Can you see my slide?
Yes, yeah, Prof. Gandhi Sensei, is all good. So you please change it to the slide view mode. Okay, yeah. So there is a loss icon. You can press that, the slide view mode.
Okay.
Yeah, currently it's like a preview mode. You can change to
not work. So
okay, at the bottom, at the right side, the bottom, the lost icon. So where you see this slide view mode. Yeah, good. So yeah, all good.
not work. So try.
Putani Sensei, you please
Yeah.
use share Windows option. That's the best one. The share entire screen, that's the best option. After that, you just put it in presentation mode. It's fine.
Yes, I share my screen, but
yeah, but it's not in slide view, because you have other slides also shown there.
Oh.
But why don't you click on the?
In my computer, I make a slide show, but it's not.
Yeah, can you try the icon at the right side, the bottom? So where this percentage That's below that there is a slide view button. If you click on that, it will change easy. Yeah.
Right?
If you, no, the right side, bottom, the bottom, not there, the bottom side.
Yeah. Okay.
Yeah, otherwise you can continue it this way. It's okay, not bad. So let us get started. It's
Okay,
fine.
sorry for some trouble. Okay, thank you very much again. So first of all, I would like to say thanks to the organizing committee to give me an opportunity to talk as a first keynote speaker in this Microplastics 2021 conference. So since this famous picture is uploaded on the internet, plastic pollution has received much concern worldwide. In particular, smaller debris, such as microplastics, are recognized as a severe problem because of possible accumulation of organic pollutant and then affecting the water environment and finally the human health. So to solve this microplastic problem, it is very important to analyze environmental microplastics. This is not work, so. Okay. So this slide shows the typical analytical procedure for the marine microplastics. In this case, surface water is gathered by this plankton net, and collected species are separated manually, and microplastics are characterized by micro FTIR and Raman spectroscopy. This graph shows the size distribution of microplastics on the marine water surface determined by the ordinary analytical procedure. In this case, observed floating amounts of microplastics on the marine water surface is much less than that estimated from eluting amounts, especially for the smaller species. So this phenomena is coming from some missing things. So possible origin of this missing thing problem is one is the because of too much fragmentation, the plastic species getting too much to detect, especially for the nanoplastics levels. And second possibility is that such smaller species are sinking into the bottom sediment, and it is very difficult to isolate them. And also some of the microplastics could be ingested by some organisms. So in the case of smaller plastics, even by the smaller organisms can ingest it. For this problem, for the missing things, the analytical pyrolysis technique is very useful because of its high sensitivity and high selectivity. So the advantage of the Py-GCMS method for microplastic analysis, even for the nanoplastic level particles, this technique can identify and quantify them. And it is not necessarily to isolate them. So mixed sample can be also analyzed. So today I show you our recent basic approach to solve this missing sim problem by using Py-GCMS. The first topic is challenging to analyze the smaller microplastic species. First topic is the identification of polymer species in complex mixture by using Py-GC atmospheric pressure chemical ionization time of flight mass spec. So Py-GCMS measurements are useful even for the nano and smaller microplastics. But even by using this method, it is important to, this is impossible to analyze individual species. So complex mixture samples should be analyzed. So in this case, reliable extraction of information of each plastic component in mixed plastic samples is necessary by Py-GCMS measurement. So we are using a very sophisticated instrument. It's a Py-GC APCI time of flight mass spec. We call this Py-APGCMS. In this case, ionization of the separated species are achieved by atmospheric pressure chemical ionization. By this ionization, molecular and molecular-related ions formed, along with some unique fragment ion. And then the ionized species are analyzed by using the very high resolution time of flight mass spec to obtain precise mass information. So here you can see the typical pyrogram of the plastic, in this case, pyrogram of the polypropylene standard sample by using Py-APGCMS. So in this case, you can see the various products in the time total ion current chromatogram. And typical mass spectra by APCI is shown here for peak 1 and peak 2. In this case, you can see the molecular and molecular-related peaks. And also, you can see the very unique fragment ions at M-Bazi 156.139, commonly observed in peak 1 and 2. And also, this unique fragment ion is observed for every pyrrolate for the polypropylene. So we focused on this unique fragment ion. We try to analyze and extract the information of the polypropylene from this mixed plastic sample. This mixed sample is a mixture of the 5 major plastics produced in Japan and mixed in this production ratio and milled into fine powder and then subjected to the Py-APGCMS measurement. So here, this slide shows the extracted ion chromatograms of this mixed sample and compared with that of the standard polypropylene sample. So you can see both pyrograms, extracted ion pyrograms are very similar, even for the mixed sample, without any interference of other coexistence plastic species. So from this paragraph, you can easily identify the polypropylene existing in this mixed plastic sample. Basically, in the similar manner, by focusing on the each specific fragment ions for each plastic species, we can extract the information of each plastic component in the mixed samples. And finally, we applied to analyze the environmental microplastic samples by using this method. This sample is environmental microplastics collected at the ocean near Japan. And 200 species are collected in this container. And then all of the species are cryo-milled into fine powder to form a model of the mixed environmental microplastic sample. And then this sample is subjected to the Py-GCMS measurements. So this right side shows the observed parallels focusing on the specific fragment ions for polypropylene, polyethylene, and polyvinyl chloride. So these paragraphs for the microplastic sample is almost similar for each standard plastic sample. So from this data, we can identify these three plastics are existing in this microplastic sample. So by using Py-GCMS method, even for the mixed plastic samples, we can easily extract the information of each plastic species. The second topic is the microplastics analysis ingested by smaller organisms. So this picture shows microplastics ingested in a zooplankton. But in this case, this microplastic is fluorescence label. So as for the ordinary non-fluorescence microplastic, it is very difficult to detect the ingested ones. And also, because of the small size of this plankton, it is impossible to isolate microplastics from this plankton body. So we need to analyze the plankton body with the microplastics. So in previous report, we have tried to analyze one zooplankton individual by Py-GC. In this case, not focusing on the microplastics, but focusing on the lipid component. But by using this technique, we try to analyze microplastics ingested by individual zooplankton. So in this case, we achieved a model experiment for polystyrene microspheres ingested in Daphnia magna by using Py-GCMS. So we cultured the model Daphnia magna samples under 4 different conditions. One is the control condition without feeding any microplastics. The second condition, Daphnia magna is cultured with polystyrene microspheres. And the third case, cultured with polystyrene and chlorella. Chlorella is the ordinary food of the Daphnia. And in this case, only the flavor of the chloride is added. And fourth case, Daphnia is cultured with polystyrene and ordinary food chloride. And individual Daphnia is cultured in separately in the cells of this microplate under 4 different conditions. And in each condition, 3 Daphnia is cultured and then analyzed by Py-GCMS. Before analyzing the model Daphnia sample, at first we prepare the calibration lines for the determination of polystyrene microsphere. In this case, various contents of the polystyrene microsphere and as the plankton body matrix, the commercially available Daphnia powder and internal standard for the determination is added in the sample cup and then subjected in the Py-GCMS measurement. In this case, we use the ordinary Py-GCMS system, not the Py-GCMS system. In this case, usual quadruple AMS is utilized, and the mixed sample is pyrolyzed at 600 degrees Celsius. This slide shows the typical pyrogram of the mixed samples for calibration, along with those for each component. So, in the large amount of the polystyrene, in this case, among the various products from the Daphnia powder, you can see clearly the styrene monomer and styrene primer from the polystyrene microsphere, like this. But in the case of smaller polystyrene content, in the case of total ion chromatogram, the monomer peak is getting much smaller. And as for the trimer peak, it is difficult to observe because of the interference of the other products from the matrix. So in this case, we use the selected ion monitoring measurement by focusing on the specific ions for styrene monomer, trimer, and the internal standard. In this case, in the same level of the polystyrene content, you can see clearly the styrene monomer peak and internal standard. And even for the trimer peaks, you can see very small but clearly observed like this. So from this measurement, we make a calibration for the polystyrene microsphere ingested in zooplankton. This is a obtained calibration line focusing on styrene monomer and styrene trimer. In both cases, a very wide range of the polystyrene content. You can see linear relationships observed with a fairly good reproducibility. So by using this calibration information, then we tried to determine the content, polystyrene content in individual Daphnia samples. In this case, individual Daphnia sample is pyrolyzed under the same condition for calibration and observe the specific pyrogram. So this slide shows the typical pyrogram for the individual Daphnia samples under 4 different cultures under different 4 conditions. as for the control samples, in this case, polystyrene microsphere was not added, but even in this case, you can see very small peak of styrene monomer. This might be coming from the Daphnia body. So styrene monomer is not suitable for the determination for the smaller polystyrene content case. So we focus on the styrene trimer peak. This is not observed in the control sample. So from the relative intensity of styrene trimer peak, we determine the polystyrene content ingested in the Daphnia individuals. So under three conditions, you can see styrene trimer peaks like this. And from the relative intensity to the internal standard, and using the calibration information, we determine the ingested amounts of the polystyrene microsphere. So in the case of second and third bands, these planktons are cultured without real food. In that case, many polystyrene microsphere were ingested, about several thousand to more than 10,000 particles. But in the case of the cultured with microplastics and the ordinary food chlorella, the ingested microplastic content is much smaller. In this case, only about 200 and 450. It is very important, interesting result to show the ingesting nature of the microplastics for this Daphnia magna. So in conclusion of the second topics, the Daphnia magna ingests the polystyrene, but in the case coexisting the ordinary food chlorella, the ingesting amount is much smaller compared with that cultured without ordinary food. This result is very interesting, and it suggests that this Daphnia magna preferably ingests the ordinary food rather than microplastics. So these experiments made for using the model culture sample, but now we try to analyze the real environmental zooplankton samples and then try to analyze the ingested microplastics. So I hope in the near future the observed results are presented. Thank you very much for your kind attention.
Your microphone is not on.
Prof, we know your microphone is on mute. Please unmute yourself. No, I think you are still on mute. Yeah, that's I think you have to unmute your microphone. Okay, I think I still has some problems. Maybe I will read the question on his behalf. Okay, we have few questions here. Okay, Gani-sensei, the first question is, how does PP alone and mixture of plastics have similar pyrogram? I mean, in the later case, why the additional peaks corresponding to the other polymer types are missing?
So in this case, we use a very sophisticated MS system, especially for the very high resolution time of flight mass spec. So the pyrrolate from other plastics has a different precise MS number. So we select only the pyrrolate from polypropylene can be observed without interfering with other species. But some species from polyethylene is also observed in the pyrrolate. So
Okay, yeah, thank you. I think so that you have used the time of flight for your screening, right? That could be reason. Dr. Vino, is your microphone okay? I think I'm still seeing muted. I think it's still on mute, so I don't know. Maybe you can leave the panel and join again. Okay, can I read the second one? Okay, there's a question on this major impacts met by Daphnia in analysis. So, can you read that also, because it is in the Q&A box? Yeah, if it is not clear, maybe we can discuss during the panel discussion. Okay, maybe the question is not clear to you, so we will ask them again. Okay, so maybe they can come on stage during the panel discussion, then we will try to address that. Okay. Yeah, so we will have one more question before we move on to the second talk. The next question is, how many marker ions of a particular polymer needs to be considered for the confirmation of polymer, as from some environmental samples also contain the same marker ion of same polymer?
It depends on the plastic species, and in the case of polypropylene, we have luckily find common specific unique ions, fragment ions. So we focus on this ion. But in the case of another plastics, we need several ions should be selected. It depends on the plastic species.
So the number of marker ions can vary, right, depending upon the type of polymer. Is that correct? Yeah. So that's like a couple more questions. So one is is there any monomer release from polystyrene as part of the screening? Do you see any monomer from a polystyrene that is released? That's the question.
So styrene monomer have possibility to coming from various sample components. So
it will be difficult to say that it is coming from the polystyrene microplastic. Okay, so I think there's one more last question. Maybe we can take it a short one. So please let me know one more time the particle size of polystyrene particle.
in this model experiment, the polyester particle size is 10 micrometer diameter.
Okay. 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 So thank you, OHTANI Sensei, for your wonderful presentation, and also there were like good questions asked by the audience. So the one of the question which we were not clear now, we will try to address it during the panel discussion. So thank you very much.
Thank you very much.
Okay. Vinu-san, how is your microphone? Because I am still seeing you on mute. Yeah, I think somehow your microphone it's still on mute for some reason, so we cannot figure that out. Okay, I think we will request the next speaker, Prof. Jas.